Dear students, for class 12th, biotechnology and its applications, the topic allotted for our discussion today by the syllabus is insulin. Production of insulin by recombinant DNA technology or genetic engineering. This topic is one of the important core topics in genetic engineering. So far as insulin is concerned, insulin is a hormone secreted by the beta cells of the pancreas. The another hormone is glucagon that is secreted by the alpha cells of the pancreas. The insulin lowers the blood sugar level, and the glucagon increases the blood sugar level. Whenever there is deficiency of glucagon, the blood sugar level decreases further than the normal level, causing a disease. That disease is called as hypoglycemia, low blood sugar level. But whenever there is deficiency of insulin, genetically or physiologically, that leads to increase in the level of blood sugar level that causes a disease that is called as hyperglycemia, or increased blood sugar level, diabetes mellitus. But so far the applications of biotechnology, especially the applications of genetic engineering, are concerned, the insulin has been prepared genetically and introduced into the individuals with the defective insulin production due to the genetic defect, and that defect has been corrected. So far the primary structure of the primary insulin is concerned, the first protein whose amino acid sequence forming this primary structure was determined was insulin in 1954 by the scientist Frederick Sanger. This protein is formed of two polypeptide chains that is clearly visible in the pro-insulin chain in front of you. The two chains are alpha chains and beta chains, interlinked covalently by two disulfide bonds. A chain is formed of 21 amino acid residues, while B chain is formed of 30 amino acid residues. The genetically engineered insulin is a product prepared by recombinant DNA technology. How this technology works and how it is formed. Shaw P. Schaefer suggested that diabetes occurs due to failure of some islands of pancreas to secrete insulin. Earlier, insulin for diabetes was extracted from pancreas of slaughtered cattle and pigs. The process was quite tiresome and difficult, and yields of insulin was very low. In some patients, it developed allergy or other side effects to foreign protein due to immune response. In 1983, Eli Lilly, an American company, prepared two DNA sequences for A and B chains of human insulin and introduced them in plasmids of Escherichia coli, that is visible from the diagram in front of you. Extract two types of polypeptide chains and combined them by creating disulfide bonds to form human insulin called Humulin. Eli Lilly has started selling Humulin since 5th July 1983. Eli Lilly and Ranbaxy launched a new insulin project, namely Humalog, an analog of 5-6 human insulin, which is more expensive than human insulin products. Humalog is absorbed within 10 to 15 minutes, as against 30 to 60 minutes of other insulin products. Other therapeutic proteins produced by microbial biotechnology involving recombinant DNA technology are antiviral proteinaceous human beta interferons, a fibrolytic protein tissue plasminogen activator, and antihemophilic factor. And this production of human insulin that is diagrammatically visible to you, how it is formed of alpha chains and beta chains with disulfide bonds, how it is prepared artificially and introduced. This is one of the important branches of applications of recombinant DNA technology or genetic engineering. That's all for today.